I would like to thank the organizers for this opportunity to present Neuralstem's very, very brief summary, 5-minute brief. I will be just flashing some of these slides. They're obviously very text heavy. It's meant for you to read after the symposium or, take it home with you. We have 2 programs. One of them is a cell therapy. Our lead product is NSI-566, which I'll be mostly talking about in the next 5 minutes. The second part of it is a small molecule. It's a traditional synthetic organic molecule, orally active. It's called NSI-189. And the link between the 566, the cell therapy, to the small molecule 189 is that it shares the same regenerative medicine theme. That is, we take certain diseases, we try to stimulate the endogenous neural stem cells that the adult brain has with taking this small molecule pill every day to treat chronic diseases like depression, Alzheimer's, diabetes, brain injury, etc. And then we treat very severe neurological diseases like a stroke, spinal cord injury, ALS, by injecting the neural stem cells into the tissue to repair them physically. The mechanism of action for cell therapy is difficult to come by. So we're very fortunate to have a independent verification of this from very recent paper in Nature Medicine from a UCSD team that has done a monkey study where they intentionally cut monkey spinal cord and then injected NSI-566 cells and show that, in fact, the stem cells not only differentiate robustly into this type of neurons, human nerve cells growing in the monkey's spinal cord into the injured part, but also it synaptically integrates with the host motor neurons below the injury to complete the circuitry to then reverse the paralysis in monkeys to be able to grip their food. So that was very good. Now we have a clinical trial that's going on in chronic spinal cord injury. We're trying to demonstrate that, and we should be coming out with results pretty soon publicly. Another trial that we're in is ALS, which is a neurodegenerative disease, and here we have done 2 clinical trials in the US, and we're looking at a comparison between the stem cell treated patients versus historical patients, and we see that at 12 months, and 18 months, and especially 18 months and 24 months, we achieve a statistically significant difference in the functionality of the patients measured by a functional scale called ALS-FRS. We also in a chronic spinal cord injury where we're trying to reverse the paralysis caused by stroke event. This is in patient population who are minimally at least about 5 months at the time of surgery to 24 months. And by adding the 9 subjects we have completed over 12 months, you can see the improvement of motor score. This is a motor scale called Fugl-Meyer motor score, and shows statistically significant improvement at basically every time we measure it, starting from 1 month, which is long lasting. 10 points is thought to be clinically meaningful improvement, and we achieve about 15 points. So that's my 5-minute cap. Thank you very much for your attention.
Great. So I guess the first question I want to ask you, Karl, is talk about your specific stem cell and what advantages it may have over perhaps the mesenchymal types of stem cells, the human embryonic derived stem cells, or even something like multi-stem.
Yeah, sure. I think that's a key differentiator. So the origin of cells, speaking of stem cells, does matter. Our cells are obtained from the developing nervous system. What that means is the stem cells are not induced, but they are naturally occurring. And in the developing nervous system, it's naturally occurring in a cell layer called the neural epithelium. And during development, these epithelium cells, which are where the neural stem cells reside, they proliferate and differentiate, give rise to adult brain and spinal cord. Our platform technology is the methodology, and as well as the composition of matter, to be able to isolate those cells and keep them in culture as multipotent neural stem cells. When we put those cells back into the tissue, or in fact in culture as well, they only differentiate into nervous system cells. Neurons, astrocytes, and oligodendrocytes. So in other words, we obtain the developing cells at the developing stage of the neuroepithelium. We put them into the adult brain that's damaged to fix the nervous system. So they come from the nervous system, they reside in the nervous system, so it's a naturally integrating cells.
Okay, so for your lead compound, and let's start with your ischemic stroke study, can you highlight any differences in either the types of patients you're treating or any other differences in treatment protocol between your study and what Athersys is doing?
Yeah, so as I highlighted, we have 3 different clinical programs right now with NSI 566, stroke, spinal cord injury, ALS. In all of those cases, the unifying theme is to reverse paralysis, to be able to reverse the motor deficit caused by each of these indications. And the reason is that the 566 cells, which are derived from spinal cord, developing spinal cord, has this tremendous property to differentiate into nerve cells, neurons. And of course, the neurons is what communicates and conducts electricity and makes be able to body move, right? And that's what gets damaged. So we are able to cause these cells to be, once transplanted, to bridge that broken connection. And so to demonstrate that, we go to chronic population who have a stable, neurologically stably deficit. So we look at chronic spinal cord injury, chronic stroke, chronic ALS that already has impairment in the motor deficit, as opposed to acute cases like in Athersys, 1 to 2 days after stroke. I think that's, or spinal cord injury, so we're looking at chronic patients who have a flat line, baseline, requires very few patients to demonstrate a change.
So for your stroke study, what's the next data release you'll be sharing with us?
We're hoping to have announcement of our data pretty soon in the next few months. Yeah.
Okay, great. And for cervical spinal cord injury, you're now enrolling the patients in the next arm of that study. And again, talk about, obviously you mentioned it's more of the chronic post-injury patients that you're looking at, but can you further define that and also the size of the dose of the cells that you're injecting?
Yes. So great question. Both in whether we're talking about ischemic injury in the brain or traumatic injury in the spinal cord or traumatic injury in the brain like a TBI, what we're talking about is a preventive, one possible window of intervention, of course, is at a very acute stage post-injury to be able to minimize the secondary damage that is oncoming to the tissue. But as I said earlier, our approach has been to demonstrate on a stable basis, and the mechanism of action that's involved in those 2 different windows of intervention are completely different. So in the acute stage, what we're talking about is a neuroprotective strategy or anti-inflammatory strategy to be able to minimize or rescue those cells that otherwise would be dead in the next ensuing time period of a month or even many months in the stroke case, as opposed to, in our case, to take already a tissue that has gone through all of that and really regenerate. And NSI-566 has shown this remarkable ability to be able to do that.
So in this current cohort, how many patients will you enroll, and when do you think we might see some interim data from that cohort?
Yeah, so we've done 4 paraplegic patients. These are completely paraplegic, wheelchair.
These are Asia A.
Asia A. There's no sensory, no motor below injury. We've done 4 thoracic injury patients. UCSD will be publishing data very soon. In the next few months, I hope it will come out, and we're seeing really encouraging signs there. And that's why we're moving forward with quadriplegic patients, because it's hard to measure changes in the walking or abdominal muscle changes as opposed to cervical change, whereas in completely paraplegic neck down, any small millimeter improvement in that reconnection can mean a huge difference in the quality of life of patients.
Okay, and what's the next step for the ALS program?
Yeah, so ALS, we have done dose escalation study in terms of number of injections, risk profile of the patient population in phase 1 and subsequently phase 2. So we are ready for a pivotal trial. We are looking for FDA's guidance on how to design that, because ALS is a very unique situation, especially with our cell therapy, which is a very invasive treatment, effective but invasive, and how to have a comparison arm is a very tricky operational issue. So we're really looking for FDA's guidance on that, and that's what we're waiting on.
And do you have a time frame for when you might commence such a next phase study?
Yeah, so for stroke, we are all ready to go. We've done 9 patients in chronic stroke patients, and we've seen very encouraging results. We'll be publishing that shortly, as I mentioned earlier, and we're right now actively designing to kick off our phase 2/3 study.
And how about for ALS?
For ALS, as I said, I think hopefully in the second half of the year, we will have a meeting with the FDA and get clear guidance on who the comparator arm should be. As I showed in the previous slide, our preference is a historical data set, but obviously that has some caveats involved using historical data set as a comparator arm. So so we're looking for FDA's guidance on that, but hopefully in the second half of this year.
Okay, well, very good. Appreciate it.
Thank you very much.